Just confirm me, am I audible to you? Good morning. Just confirm me, am I audible to you? Okay. Let's wait for 1 minute.
Recording.
Okay. What we have saw yesterday is Helminthes, and we have completed one of the parasite, that is Tenias. What is the common name for Tenias? Just tell me. What is the common name for Tenias? Quick. Tapeworm. Very good. Okay, what is the other name of blader worm? What is the other name for blader worm? What is the other name for blader worm? Very good. Cysticercus. Clear? Okay. And this Cysticercus is the one of the stage which has been involved in the life cycle of the Taenius oleum. Don't forget, okay? Now, and what are all the hosts? What is the primary host and what is the secondary host for this Taenius oleum? Primary host and secondary host involved in the life cycle of tapeworm is very good. Man is the primary host and pig is the secondary or the intermediate host. Very good. And as it is involved in the two hosts, it is digenetic in nature because it needs two hosts to complete their life cycle. Okay, that is about Taenius oleum. In today's class, we are going to see about cystosoma. Okay, fine. See, first question you have to remember is what is the common name for cystosoma is blood fluke. Okay, now this blood fluke of man and other animals are placed in the genus Cystosoma. So this genus is completely referred to the blood flukes, those who feed on the blood. Cystosoma, it is also called as a bilharzia, and why it is said to be as bilharzia means it is discovered by the bilhars in 1851, and these are all the three species which are going to infect the man. And what is the three species? Cystosomia hematobium, Cystosoma manzoni, and Cystosoma japonicum. See, Cystosoma hematobium. See, the name suggests hem means blood. So it is going to inhabit in the blood vessels of the bladder and the urinary tract of the human. In case of Cystosoma manzoni, it is present in the small branches of mesenteric or portal veins. In case of Cystosoma japonicum, it is present in the small veins of portal and the mesenteric vessels. This cystosoma is dioecious in nature. What is dioecious means? Male and female reproductive organ which is present in the different individual. That means sexes or separate. They are present in the separate individual. Okay, dioecious is unisexual in nature. Okay, don't forget this. Very good. Separate. And here with well-defined sexual dimorphism and the two intestinal limbs becomes united into one at the posterior end of the body. Here this female is more slender and cylindrical and lies permanently lodged in the gynocorpic canal of the male. See, you have to remember throughout their life cycle, this male and the female are completely coexist together. See, of course, there is a separate individual. There is a separate individual of cystosoma, male and female. Okay. But this female and male or made in contact with each other throughout their life cycle. Okay, female is more slender and cylindrical and it lies permanently lodged in the gynocorpic canal of the male. Okay, this is the name of the canal present in the male by which the female is attached to the male. Okay, formed by the rolling of sides of body behind the ventral sucker. You have to understand this male and female are coexisting together. It is placed under the phylum Platyhelminthes. It is placed under the class Trematoda. Trematoda means it is again dorso-ventrally flattened. It is leaf-like in shape. It is in leaf-like shape. Okay, that is those parasites will be placed under the trematodes, and those parasites which is placed under the trematodes have two suckers. One oral and one ventral. See, two suckers is present, and that is one in oral and one ventral inside, and the order protostomata. And what does protostomata mean? This comprises the trematode worms with the mouth or near the anterior, with the mouth at or the near the anterior end of the body. The mouth is present at the anterior end of the body. That is why it is said to be as protostomata, and it is placed under the family Cystostomatidae. What is the family Cystostomatidae means? This is the family of digenetic trematodes. Okay, digenetic, which means it requires two hosts. Digenetic trematodes with complex parasitic life cycle. Those parasites are placed under the Cystostomatidae. And again, there are three different species, right? We have seen Cystosoma hematobium, manzoni, and japonicum. But here we are going to see about Cystosoma manzoni. Okay, and first information you have to remember is this Cystosoma manzoni is digenetic parasite. Clear? And this life cycle has been completed in two hosts. And the primary host is human and the secondary host is snail. Clear? See, this will be your questionable area. This is a digenetic parasite and it completed a life cycle in two hosts. And the primary host is human, secondary host is snake. And you have to compare all the parasites with their host. In case of Taenia solium, what we have studied is their primary host is human and the secondary host is pig. But here in Cystosoma manzoni, the primary host is human and the secondary host is snake. This will be your questionable area. And first thing, mating and egg production. How they are going to produce eggs. See, this is how it look like. This is female and this is male. Okay, see how the female is lodged in the male through the gynecophoric canal. This is the name of the canal. See, the male worm form a characteristic groove. This name of the groove is said to be as a gynecophoric canal within which resides the adult female parasite. Okay, from where the female parasite attached with the male. So thus appearing as a characteristics couple. Although it is unusual to have a separate sexes for trematodes, but this can be explained by the fact that female worms lives permanent relationship with the males. See, as I said before, both have a different individuals. Their sexes are separate, but they have been in a contact relationship permanently. They contact with each other permanently throughout their life cycle. Clear? And after mating, the coupled worms migrates against the flow of blood and finally reaches the small venules of the intestine. That is, intestine is said to be as the mesenteries, particularly the mesentery circulation. See, after mating, this coupled worm migrates. We have to know that cystosoma is a blood fluke, right? We have know that the cystosoma is a blood fluke. So obviously they are going to feed on the blood. So they are going to migrate against the flow of blood. They migrate along with the blood, and finally, where it's going to reach? It reaches the intestine. How the eggs of Cystosoma manzoni look like means light brownish brown. The length is not necessary. It is elongated, and there is no operculum is present. Operculum is nothing but the opening which cover the opening has been covered by a covering called operculum, and characterized by possessing a large backward pointing lateral spine revealed under the stem and the electron microscopy. See the cecal region. Cecal region means what? It cecum means one of the part of the large intestine. Cecum region and the large intestine region of the worm is surrounded by the small inferior mesenteric vesicles which act as a final niche for the paired worm. So what is the exact location through finally the cystosoma, the targeted area of cystosoma is this large intestine. The mature female Cystosoma manzoni lays approximately 300 eggs per day, deposited on the venous capillaries endothelial linings. From the intestine, see that from the endothelial lining of the egg Cystosoma manzoni moves into the lumen of the intestine and excreted out into the environment with the host feces. Obviously, it is going to reach the intestine, and how they are going to reach the external environment means through the host fecal matter. And as soon as the egg of the cystosoma manzoni is emitted into the water, now where it is going to, it has to reach the water. Okay, through the fecal route, it has to reach the water. From there, water is necessary for those eggs to hatch out into the merycelium. This will be the questionable area. See, what are all the points I'm going to say now? Just take down. See, the first thing is common name, blood fluke. Cystosoma is said to be as a blood fluke. Clear? And the second point is, see these three species, the name of these three species and their inhabitations. That is the main thing. And the third thing is, this is the second thing. And the third thing is dioecious species. Dioecious, and this fourth, female and male are completely lodged in the gynecophoric canal of the male. Okay, these four things you have to remember. And the fifth one is digenetic in nature. Cystosoma manzoni is the digenetic in nature. It needs two hosts to complete their life cycle. And the sixth point you have to remember is what are those hosts. And the primary host is human and the secondary host is snake. And these are not that much important. Now these things, see, after it has been introduced into the human, that is primary host, all the eggs will be released out into the human fecal matter, and these via this fecal matter, the eggs which is present in the human fecal matter, it has to reach the water. Where it has to reach, it needs water. In that water, the eggs will hatch out. Okay, in the water, the eggs will hatch out, and what the eggs gives rise to? The egg will give rise to the merycelium. Okay, this is the most important things. This is the seventh important point you have to keep in mind. The eggs has to reach the water from the human fecal matter. If it didn't find any water, means the egg cannot hatch out and it cannot produce a merycelium. Dilution of feces with the water, light and temperature are the factor influencing the hatching of eggs. So temperature is also the main point to hatch out those eggs into the merycelium. Okay, this is the seventh important point you have to remember. Now, what is merycelium? The matured merycelium moves through the water as the outer surface is covered with numerous cilia. So you have to remember this merycelium is motile. And how it can be moved from one place to another means with the help of a locomotory organ called the cilia throughout their entire organisms. So you have to remember. So the eighth point you have to remember is this merycelium is motile and it has a cilia in it. And it has an outer covering, has a 4 layers of epidermal plates, and at the apex of merycelium, ciliated sensory organelles, epica, papilla, and the number of gland cells are present. These are all the characteristic features of those larval stage, that is merycelium. And Terebratorium at its apex consists of number of membranous folds and helps in its instant attachment to its intermediate stale host prior to the penetration. So what is the name of that structure which helps the merycelium to attach with the intermediate host, that is nothing but the secondary host, that is snail, is Trebatorium. Trebatorium is a structure like the suckers and hooks present in the Taenia solium. Trebatorium is present in the cystosoma. This will helps the cystosoma species to attach with the intermediate host or the primary host or the snail. Clear? So the ninth important point you have to keep in mind is this point, tribatorium. Clear? So you have to remember the meracidium is motile because of cilia. And through this water, it is going to reach the intermediate snail, intermediate host. Okay, fine. Next, this meracidium has a variety of sensory organs connected to a nervous system. So it has a sensory organ so that it can sense the intermediate host. It has a pair of posterior flame cells. What is flame cells and what is their function? What is flame cells? Kindly please be regular to the class and please be on time. What is their very good excretion? Flame cells, it is present only in the phylum. Flame cells is present only in the phylum. This can also be asked in the question. Platyhelminthes. And what is their function? Excretion is their only function. Clear? So this is the most important point when it comes to the phylum Platyhelminthes. As the cystosoma comes under the phylum Platyhelminthes, obviously the excretory organ or the excretory cells present in this cystosoma is flame cells. And it has a pair of posterior flame cells and a pair of anterior flame cells constituting the excretory system. These are sexually differentiated forms of a parasite. The energy required by the meracidium takes 4 to 6 hours to search up appropriate fresh water and the snail. See, it is a human fecal matter. From the fecal matter, the eggs will hatch out in the water. So the egg now hatch out in the water. Now meracidium will be produced. Now this meracidium have to reach to the snail. Okay, to reach to the snail, it has to move from one place to another so that it has what is that locomotory organ cilia, and it has a sensory organs and it has a excretory system that is flame cells. So everything will be present in the meracidium. These are all the things you have to keep in mind. The sexually differentiated form of a parasite. Now, what are all the intermediate host for the meracidium? So you have to remember the snail species. If they give, if they in the examination, if they give their snail means there is no problem. You are easily answer those questions. But if they give the scientific name means you will get struggle. Okay, Biompleria, Straminea, and Tinagophila and Glabrata. These are all the species of snails. So you have to remember the name of the species, at least the genus names. Okay, genus names is very important, but you have to remember the species names. Okay, fine. Snails comes under which phylum? Snail comes under which phylum? Snail comes under which phylum? Very good. Mollusca. Okay. Phyla globosa, we are going to study about those phylums clearly. So just remember this snails comes under the phylum Mollusca, and these are all the species of finals through which the meracidium will reach these phylum species, these snail species. Okay. So as I explained when I explained about the syllabus, what are the things I have explained is the larval stages. Okay, the larval stages is very important. So the first larval stage is meracidium, and after the meracidium, meracidium is going to convert into the cercaria. Okay, see, just make a flow chart. This is the human fecal matter. Now the in the human fecal matter, the lots of eggs will be released in those fecal matter. Now it has to reach the water. The eggs will hatch out. Now, in that water, the meracidium will be produced, and meracidium will reach the snails. And after that, this meracidium will be converted their structure into the cercaria. Clear? Now, cercaria, an infective larval form of schistosoma, characteristically has a bifurcated end and long tail. So this is the characteristic features of cercaria. It has a two bifurcated tail. Now, the outer surface of the tegument is covered with the glycocalyx, furnished with sensory papillae and the spines. It is also covered with 3 layers of the plasma membrane. As it has the 3 layers of plasma membrane, it is said to be as the trilaminate. Glycocalyx, which covers the outer surface of calcarea, is a sugar-rich layer. Okay, we know that glycocalyx is present not only in this particular parasite, it is present in the capsule of bacteria, and that capsule is made up of glycocalyx. And glycocalyx is nothing but a sugar-rich layer. And the tegument of the Schistosoma manzoni characteristically has a prominent backwardly positioned spines. A number of ciliated sensory papillae are found on the cercaria. See, ciliated sensory papillae, it is present in the cercaria. The longitudinal muscle situated below the external layer of circular muscles that interserve with diagonal fibers and thus from the body walls musculature of the cercaria. This is not that much important. What are all the points is remember is important when it comes to cercaria is it has two tail, and after the mycelium cercaria will be evolved as a process of metamorphosis. This is the only thing you have to remember. And another one is it has a sensory papillae and the spines within it, and it has a 3 layers of plasma membrane. Okay, again, ciliated sensory papillae. It is present in the cercaria. Okay, these are all the things you have to remember. And another point is it is possessed by its head is possessed by 5 pairs of acetabular glands, which are the prominent feature of larval anatomy and comprise of 2 pairs of pre-acetabular and 3 pairs of post-acetabular glands. And what is the function of these acetabular glands will be discussed in the upcoming slides. Okay, this is about cercaria. When it comes to cercaria, you have to remember two tail. You have to remember about the tail and what are they made up of: glycocalyx and sensory papillae and spines. And cilia is present there. And you have to know about the acetabular glands. Okay, that's all about cercaria. And another thing is cercaria is the stage will be emerged after the stage mycelium. Okay. Now, these glands are unicellular in nature. What is this? Acetabular glands are unicellular in nature, protrude through the cone at the anterior end. These unicellular glands has role in their host adhesion and penetration. So, we have studied about the penetration glands in the taenius oleum. Okay, like in the same way, this acetabular glands also act as the penetration glands to attach with the host. Okay, to help the organism, to help the parasite to move from one place to another inside the body of the host. That is the function of those acetabular glands. Just remember, this acetabular glands is helpful in the is also can be considered as a penetration glands, suggested by the mucus-like secretion from the post-acetabular glands and proteinous secretion from the pre-acetabular gland. This is the thing you have to remember. We have studied about the acetabular glands. In that, we have two types of acetabular glands, that is post-acetabular and the pre-acetabular. This post-acetabular glands will secrete the secretion, mucus secretion, through which it can be easily passed from one place to another. In case of pre-acetabular glands, it is a proteinaceous secretion to digest some of the enzymes present in the host. Moreover, at the anterior end, they also possess the unicellular gland called the head glands. Okay, again, this acetabular glands is also an unicellular gland, and this head glands is also an unicellular glands, which provides membrane material needed during the development of cercaria to schistosomula form following the host penetration. Now, this cercaria characteristically possess an anterodorsal pair, a ventral and posterior dorsal pair, mid-ventral pair, a pair of flame cells in the anterior tail. Those total have 5 pairs of flame cells. This is the characteristic features of cercaria. A tightly packed ciliary roselets occupy a region at the base of the flame cells. The first tubule cells and the flame cells are interdigited to form a basket or a barrel-like structure, form a ciliary tuft of flame cells. So you have to remember cilia plays a major role when it comes to the mycelium as well as the cercaria. So these two stages are the most important stage of the this parasite, schistosoma, and how this infection have been occurring. The Meracidium stage is the one which has been introduced into the snail host. Okay, once it has been entered into the snail host, it undergoes movement over the snail's food surface to reach its preferred sites of penetration. See, Meracidium is the name of the stage. After hatching from the egg, it has to reach the snail. Okay, after it reaches the snail, it has to move the body and move inside the body where it can find the exact location to further metamorphosis. And exploratory movements are followed by the boring movements of the Meracidium. What is exploratory movements and the boring movement means? Exploratory movement means finding the place to which they can undergo the upcoming metamorphosis process. Followed by boring movements means after finding the process, they are digging to the host. Okay. Boring movements of the Meracidium and trigger the secretion of proteolytic enzymes from the gland cells to perform the penetration. It has to circulate inside the body of the snail. For that, it needs some of the secretion. For that secretion, penetration glands is going to help, and that is nothing but acetabular glands, post and pre-acetabular glands. One is going to secrete the mucus secretion and another is going to secrete the proteolytic secretion. Now the muscle layers disappears and the ciliated epidermal plates are shed as the meracidium penetrates into the intermediate host and transformed into the primary sporocyst. This is the question here. See, the meracidium penetrates into the intermediate host and transforms into the primary sporocytes. Primary sporocyst. Thus, this meracidium develops into the sporocyst inside the body of the snail host. Just remember this point. So the ninth important point you have to remember is the meracidium develops into the sporocyst. And what is happening? See, meracidium will be present outside the body of the host. Okay, now this meracidium is entering into the pig. Inside the body of the host, this meracidium will be converted into the sporocyst. This is the point you have to remember. Okay, now the mother and daughter sporocyst or the two generation of the sporocyst. Now inside the body of the snail, they are multiplying. They are getting multiplying, and we can call them as a mother and daughter sporocyst. That is the two generation of the sporocyst. It's the daughter sporocyst which produce the cercaria. Okay. See, this is the meracidium. Meracidium will be enter into the pig. There they can be converted into the sporocyst. The sporocyst we can tell as a mother and the daughter sporocyst. This daughter sporocyst, the one gives rise to the cercaria. Clear? So the question may be asked in the examination, but I have explained this meracidium is converted into the cercaria. Means what I'm trying to explain is cercaria is the stage after the stage of meracidium. But what are all the intermediate stages have been taking places? This meracidium is converted into the sporocyst. Particularly the daughter sporocyst is the one converting into the cercaria. And this cercaria is the thing, snail. Okay, snail. I'm sorry. See, this meracidium is entering into the intermediate host, that is snail. Okay, after introducing into the body of the snail, it has been converting their structure from the meracidium to the sporocyst. Okay, the sporocyst undergoing the division. We can call them as a mother sporocyst and daughter sporocyst. Now, this daughter sporocyst is the one giving rise to the cercaria. And this cercaria is an infective larval stage. This is the one you have to remember as the 10th important point. This cercaria is an infective larval form which is released into the water. Now, what is the thing? From the water entering into the snail is meracidium. And from the body of the snail, again the larval stages is entering into the water body so that it can reach the another primary host, that is human. Okay, and what is this larval stage is cercaria, and this is an infective larval form. There is no radial stages is present here. Generally, meracidium, sporocyst, cercaria, after the cercaria, radial stages will be present, but here there is no radial stages. You have to remember this order. Okay, when a man came in contact with infected water carrying this infectious Cercaria of the Cystosoma manzoni during bathing, wading, and washing, the host may acquire the infection. The larval form of parasite first get attached to the skin surface of the host with the help of their ventral suckers, and as the time passes and the water evaporates, the larva penetrates the skin, invade the periphery circulatory system, and infection rarely occurs on consuming the infected water. In such cases, the larval stage invade the buccal mucosa to reach the blood. See, what is the main import? These are all, these story are not and all necessary. Mycelium sporocyst, daughter sporocyst converted into the cercaria. From the water, mycelium will be entered into the body of the snail, and from the body of the snail, this cercaria is an infective larval stage. It is released into the water so that it can make into contact with the human. Okay, if the human consumes the infected water, again they are going to reach where? Blood, because this is the source of food for the Cystosoma species. Clear? Now, penetration of Cercaria into the man. Now, the penetration of infectious larval stages from the body of the snail. Okay, it has to introduce into the primary host or the definitive host. It will be occur in three phases. First one is initial attachment, and the second one is exploratory movements, and ultimately it has been penetrate into the blood vessels. Initially, the larva gets attached into the skin of the human host in context. Okay, after it has been attached with the skin of the primary host, that is human, it has been followed by the exploratory movements of the Cercaria. What is exploratory movements? They are going to dwell into this, creep over the skin surface of the host for a suitable site of penetration, such as hair follicle, further followed by final penetration of Cercaria into the skin epidermal layer. See, this considered this as a skin. Now the parasite will be present in the made in contact with the human surface. Now it has to dwell, it has to open the skin. That is said to be as an exploratory movement. And after that, it has been enter into the penetration. It is moved from the skin and enter into the body of the host. These are all the three important movements when it comes to the penetration of bacteria into the human. Okay, and how they are going to enter into the inside the body of the host means by proteolytic enzymes produced by the pre-acetabular as well as the post-acetabular gland secretion. For this penetration, the secretion is very important. And what is the gland? Name of the gland is going to secrete something is acetabular glands, particularly the pre and the post-acetabular glands. Okay, it takes nearly 30 minutes. Now, after transformation, the cystosomulum, the larval, lost its tail. Okay, we have studied this Cercaria having the tail. Now, after transforming from the body, water body to the primary host, that is human, it is going to lost its tail and shed the tegument, trilaminate plasma membrane along with the glycocalyx. Okay, everything will be destroyed and it has been replaced by matured form of the tegument. Okay, now the primary gut, that is intestine, also grows and the cystosomula invade the dermis. Dermis is nothing but the skin region. Now the parasitic form penetrates through the epidermal basement membrane to cross over in dermis. Once the cystosomulae invades the dermis, it takes about 10 hours to reach the blood vessels. It takes about 10 hours to reach the blood vessels where they get localized on the penetration. This is the main area they have to inhabit. Because cystosoma is what? Blood fluke. Once they enter the mesenteric circulation through the blood flow, the parasitic form by a pulmonary artery, it reaches the lungs. From the blood vessels, it is going to reach the lungs. These are all an extra important points, but what you have to remember is once it enters into the body, it lost its state. Also, the trilaminate plasma membrane, and it has been replaced by the matured tegument. This is the point you have to remember. And from these, they are going to reach the blood vessels, and after the blood vessels, it is going to reach the lungs. This is the only point you have to remember. Now, after it, now it has reaches the lungs, and what is there? It is going to take place in the lung. The lung state cystosomulum. After the 8th day of penetration in the lungs, the cystosomulum becomes slender and longer, attributing to a substantial increase in surface without change in the body mass. Body mass never getting changed, just their size will be increased by increasing their surface area. It is suggested that during this lung stage, the parasite starts feeding on plasma instead of blood cells of the definitive host. See, we have know that we have when we centrifuge the blood, we can get two kinds of blood. One is serum and the plasma. Okay, and this serum contains some of the things, and plasma contains some of the contents. Both are the contents of blood cells, but this particularly this cystosomum, when it's present in the lung stage, they are vigorous feeders of the plasma of the definitive host. Definitive host is nothing but a primary host. And this parasite migrates to the hepatic portal vein through the blood vessels. Finally, it reaches from the lungs to the liver. So, initially, it reaches in the blood vessels. After the blood vessels, it is going to reach the lungs, and after the lungs, it is going to reach the liver. Okay, when it is there in the lungs, it is going to feed on the plasma. These are all the things you have to remember. Now, hepatic stage of the stomula. When you heard about the names hepatic, hepatic portal vein, whatever it is, if you heard about the name hepatic or hepato, you have to remember that is deal with the liver, because the liver is made up of which cells? Hepatic cells. Now, the cystosomulum undergoes the lateral growth as it enters into the liver. Once it enters into the liver, the actual growth of the cystosoma will be happening, and the complete maturation of gonads and development of gut will be happen only when it reaches the liver. The adult female and the parasitic worm pair up once they become mature. See, once they become sexually mature, these female and male parasitic worm will getting paired with each other in the lungs and migrate against the blood flow through the hepatic portal veins to ultimately they reach their niche. What is their niche? What is their exact location to do their function as well as their habitat is mesenteric vesicles which surrounds the intestine. Okay, mesenteric vesicles means nothing but intestine. The mature form of the parasite pair and perform mating as they get localized in the mesenteric veins. See, male and female getting attached with each other when they are in the liver, but when they going to lay eggs only when they reach their niche. Niche is nothing but where their functional role is happening, and that is nothing but an intestine. When it reaches the intestine or the mesenteric vessels or veins, they are going to start laying eggs. Clear? And it takes nearly 25 to 30 days to commence eggs laying from the time of infection with the Crypteria. So, the life cycle of Schistosoma manzoni. We have this is the summary. Okay, this is the summary I have explained here. See, the contaminated water is the main source. We have studied from the contaminated water, which is the name of the stage which is going to enter into the primary host. Just tell me, contaminated water. From the contaminated water, it reaches the human host where is going to cause the infection. And what is the name of the stage? What is the name of the stage which is infective in nature, which is transformed from the water body to the human host? Very good. Cercaria. Okay. It is going to reach the definitive host. And snails is considered as the intermediate host of the parasite which produces water swimming larvae called the Cercaria that reaches their mammalian host in the water. These free-swimming larvae forms penetrate the skin of the human host and loses its tail. So this is the important characteristic features. See, once it reaches the human body, there is no purpose of tail's presence. Okay, so tails will be lost. And also the trilaminate layer of the plasma membrane. Okay, and it has been replaced by what? Matured tegument. At this point, the cercarial body changes to the cystosomulum. Okay, a migrating juvenile worm. Juvenile worm means what? Larval stage, young ones. These migrating worms enter the blood circulation system of the human body, and from blood vessels they are going to reach the lungs, and from lungs they are going to reach the tail is for swimming in water. Of course, tail is for swimming in water. Once it reaches their host, there is no need for tail. So it will be lost there. Yes, of course, it is need only when they are swimming in the water to reach the host. And see, from the blood vessels, it reaches the lungs. And from the lungs, where it is going to reach? Liver, where the sexual maturation is going to happening in the liver. Okay, male gonads and the another important characteristic developments will be happening only in the liver. Very good, liver, and matured into the adult forms, and the fusion and the attachment of this male and female or parasite will be happening in the liver. And final destiny of that parasite is where? Mesenteric venules, which is surrounding the intestine. This is the final destination of these worms, through which the female can lay many eggs. Okay. Following the skin penetration, the parasite takes about 6 weeks to reach the mesenteric venules of bowel or the rectum. Okay. These male and female feed on the blood. We know that. This is the only important thing. Other things are not important. And they are going to feed on the blood, and they are going to hatch out many eggs when they are in the intestine. Now, this eggs will be present in the intestine. It will be released by the human in the fecal matter and in the part of the urine. See, this is where a difference will be there. See, we have studied about 3 different species of cystosoma: Manzoni, Japonicum, and Hematobium. See, the eggs of Cystosoma manzoni and Japonicum are shed in feces in part in urine, whereas Cystosoma hematobium in urine partly in feces. What is the difference? What is the meaning of this statement means? With respect to this two species, Manzoni and Japonicum, majorly the eggs will be released in the feces, only some in the urine. But in the hematobium, majorly they will be released only in the urine. Only some of the eggs will be released in the fecal matter. Okay, and after it has been released from the fecal matter, now the egg has to reach where? The egg has to reach the water. In the water, the egg will be hatched out. To hatch out of this particular egg, what is the factor they are influencing to hatching of these egg means a light and the temperature. And if the light and the temperature is very favorable, if the egg find the light and temperatures is very favorable, now this egg will be converted into the mycelium in the water. Okay, this mycelium has cilia with it, and this mycelium will going to reach the snail. Okay, they are going to reach the snail, and from where this mycelium will be converted into the sporocyst. And this sporocyst, we have mother and daughter sporocyst, and the daughter, particularly the daughter sporocyst, will be converted into the cercaria. And this cercaria will be again released into the water. Okay, and again from the cercaria is an infective stage. Okay, and it has a bifurcated tail. Okay, via this tail, it is going to reach the human. Okay, once it reaches the human, it loses its tail, and further happenings will be going to happen. This is the thing you have to remember. This is the only thing you want to remember when it comes to cystosoma. Okay, and after the humans, what are all the things I have explained? It has to penetrate three types of three modes of infection will be happening. Once it wants to enter into the body of the host from the water body, it has to attach first. It has to attach first, and it has to undergo two types of movement: exploring movement and the exploratory movement and the boring movement. And after the movement, it has to penetrate inside the body of the host. These are all the three things will be happening. Once it has been entered into the body of the host, it has to reach the blood vessels, and after the blood vessels, it has to reach the lungs, and after the lungs, it has to reach the liver, and where the sexual maturation is going to happening only in the liver. And after the liver, it is going to reach the intestine. And intestine, again, the eggs will be released in the water. Then the cycle continues. This is what I have explained in this picture. Okay, if you read this picture, it will be very easy for you. Now, any doubts? If you have any doubt, means you can ask me now. or shall we solve the question? If you have no doubts, answer for this question. What is the other name for schistosomiasis? Quick. What is the other name of schistosomiasis? Bilharzia, malaria, pediculosis, thriasis. Why are you thinking much? Answer quickly. Why the thriasis? Is it? The answer will be Bilharzia because the name of the scientist who discovered this particular parasite, this particular genus blood fluke is Bilhars. Okay, so this has been also called as a Bilharzia. Clear? Now, how many species are present in the Schistosoma parasite? How many species we have studied? First and second slide. Okay, let me summarize once again for you. Please be on time, okay? So the other people also getting affected because of the repeated saying the same information, okay? So consider this point. Good. Okay, see? Cystosoma, the first thing you have to remember when it comes to cystosoma is the other name. Okay, the common name of cystosoma is blood fluke. Okay, now there are three different species which are going to infect the man. And what are all those species? Cystosoma hematobium, Cystosoma manzoni, and Cystosoma japonicum. And you have to remember where they are going to inhabit in that particular man. In case of hematobium, in the blood vessels of the bladder and urinary tract. In case of manzoni, it is in the small branches of mesenteric and portal veins. In case of cystosoma japonicum, in the small veins of portal and mesenteric vessels. Okay, these are all the three important species under the cystosoma, under the blood flukes. Okay, and another point you have to remember is this cystosoma is dioecious in nature. Dioecious means what? Male and female reproductive organ will be present in the different individual. Yes, yes, exactly. He means blood. Like in the same way, you have to remember for all other species also. And another thing is this cystosoma is dioecious in nature and it has a sexual dimorphism. Sexual dimorphism means what? Well-developed reproductive organ, and those male and female reproductive organ will be present in the separate individual, dioecious or unisexual, or the sexes are separate. Okay, both are same. Everything will be the same. Now, the female and the male always like attached with each other. Okay, female is slender cylindrical and it is completely attached with the male via the gynecophoric canal. Okay, this is the name of the canal through which the female will be attached with the male. Okay, so this point is very important. Okay, just make a note of it. Now, these are all the systematic taxonomical classification or the systematic position of the Cystosoma manzoni. See, there are three different species, but we are going to study about the Cystosoma manzoni. And you have to remember this point. The Cystosoma manzoni is digenetic in nature. Okay, so the primary host is human and the secondary host is snake. Okay, is it enough, Shanti Karunya? Okay, this is the first two slides. Let's come back to the questions. Please be on time. You may lose some of the information. Okay, there are how many species of cystosoma species? Please be quick. Answer will be 3. Yes, very good. Some of them have answered. There are 3 different species of cystosoma parasite. Okay, the penetrations is in dash in the mode of transmission for the cystosoma manzoni. The penetration is in, very good. The penetration initially it has to bore the skin. Okay, the skin will be the answer for this question. Very good. Schistosomiasis is a which type of disease? Waterborne, airborne, foodborne, and all of these. Very good. It will be the waterborne disease. Very good. Because through which the infected water, this parasite will be transferred to the human as well as a secondary host. And the intermediate host in the life cycle of schistosomiasis is, yeah, snail is an intermediate host. Good. Now, the children who are repeatedly infected with this disease can develop which of the following? If the children will be repeatedly infected with this disease, will develop which of the following? Why only anemia? See, because it is a blood fluke, it is going to consume lots of plasma in it. Okay, lots of plasma. So obviously, the children will be results in anemia. Of course, malnutrition, right? Blood carries lots of nutrients to all other parts of body. Okay, so malnutrition is also a complication. If there enough nutrition doesn't reach the brain, means obviously the brain can't concentrate in anything, so learning difficulties will also be the symptom. So this will be the answer for this question is all of the above. Clear? Fine. That's all about the schistosomiasis. Next we are going to deal with the another parasite. 1 minute. See, in Helminthes, what are all the things we have studied is Taenia solium and Schistosomiasis. Okay, in today we are going to see about the Ascaris, which is comes under the nematode category. Okay, oh, now we are going to see what is nematode and their classification and all. See the general characters of nematode. Okay, this platyhelminthes and nematodes, or the nemathelminthes or the ashelminthes, both are comes under the Helminth classification. Now, see, this is widely distributed aquatic, terrestrial, and parasitic or free living. You know that. And the body is elongated, cylindrical, unsegmented. The concept of segments will be start only from the phylum Annelida. I'm repeatedly saying the same statement. When it comes to the question in the exam, you should not get confused. The segment will be, the concept of segmentation will be arise only after the phylum or only from the phylum Annelida. Okay. Fine. Unsegmented, worm-like, and bilaterally symmetrical. We know what is bilaterally symmetrical. And again, it is triploblastic in nature and organ system grade of organization. So this is also an important one. And it has a thick cuticle, cellular or these are all the layers. Okay, these are all the layers. Thick resistant cuticle. The cuticle can have, so that as it is thick resistant cuticle, these cuticle will protect the body of the parasite. This is only for the protection. Or the syncytial epidermis, this is an outer layer, and only longitudinal muscle fibers in 4 bands. And the true coelom is absent here. Okay, but in case of platyhelminthes, what is the phylum? These are all the things. These are all the comparison. The question can be asked in the way of comparison. Okay, like I have explained. See, from what is the comparison between the phylum Porifera and the Coelenterata? In last year, in the last time, you will get a question, right? By comparing the phylum Porifera and the phylum Coelenterata, they have arised a question. Like in the same way, by comparing the platyhelminthes and nemathelminthes, you can get a question. So these are all the differences you should remember. Persistent blastocele or the pseudocele present is not lined by the mesoderm, which means what? It is a pseudocoelom. Pseudocoelom is present here. Pseudocoelom. I don't want to repeat the same thing again and again, so you'll know that. No cilia, no circulatory, and no reproductive system. Okay. See, digestive system is complete with anus. What is in case of platyhelminthes? Digestive system is incomplete because every other organ in the digestive tract is present except the anus. As anus is not present, the digestive system is incomplete in case of platyhelminthes. But in case of digestive system in the nemathelminthes, it is complete because starting from the mouth and ending with anus, all other parts of the digestive tract is present. Okay, so it is complete with muscular pharynx and non-muscular intestine. Excretory system of glandular organs or canals or both. Flame cells are absent here. See, flame cells is the only characteristic which is present only in the phylum Platyhelminthes. So it is absent in the Nemathelminthes. Okay, fine. Next. Nervous system is the circumference ring and anterior and the posterior nerves. So nervous systems is also developed here. Sensory organs is poorly developed in the form of small papillae and the amphids near on the two body ends. So the sensory organs is poorly developed, but it is primitive in nature. A simple papillae is present. See, dioecious in nature. What is dioecious? Against sexual dimorphism. Sexes are separate. It is unisexual in nature. Now, the male is smaller than female. So this is the general concept. See, not only this worms, these parasites, not all microorganisms. Generally, the main majority of the male a majority of the species has a small male but larger female. Okay, female are generally bigger in their size. Okay, because of the reproduction function. Okay, females are generally bigger in size. So the question can also be arise. Okay, the males are generally larger than female in every species. Like they can raise a question. They can throw a question like that. But you have to know this is the general concept. This is a basic understanding that males are generally smaller than the female in every species, almost all the species, not every. The gonads are simple and coiled. Male genital ducts leads to the cloaca. These are reproductive organ, and female genital ducts with a separate opening, no asexual reproduction. As the sexes are very well developed, it has a very well reproductive organ. Definitely, they are going to undergo a sexual reproduction. And what is the sexual reproduction type? Internal fertilization. Definitely fertilization is going to happen there. And what is the type of fertilization? As they are a well developed organism, definitely the fertilization is internal. The young ones is going to develop, the embryo is developed within the body of the female, and the development usually direct with or without an intermediate host. Okay, it may be a monogenetic, it may be a digenetic, but the development is direct, no larval stages. And the classification of nematodes is class 1 Ephasmidae and the Plasmidae. Ephasmidae is also called as Adenophorae, and the Phasmidia is also called as the Caesarnymphia. Okay, now see what is plasmids. If the plasmids is absent, then it is said to be as the Aphasmidia. If the plasmids is present, then it is comes under the class Plasmidae. What is this plasmid means? Caudal sensory organs. What is caudal? Caudal means a tail region. Okay, caudal fin. In the fishes, we are say caudal fin. And what is the caudal fin means? Tail fin. Okay, so this plasmids, it is said to be as a caudal sensory organs. In the tail region, there is a presence of caudal sensory organs. If that plasmid is absent, then it is placed into the class Plasmidae. If the caudal sensory organs is present, then it is comes under the class Plasmidae. Just remember the other name also. Amphids. Opposite to the plasmid, it is an anterior sense organs of various types and rarely pore-like. There is again no excretory system is present here. If present, then it is poorly developed. Mesenterial tissue is well developed. The tissue which surrounding the gut or the intestine, that is said to be as the mesenterial tissue. Coming to the class Plasmidae, amphids are pore-like, just like the Aphasmidae. Excretory system is developed. Mesenterial tissue is weakly developed. Okay, this is the opposite point as Aphasmidae. In case of Aphasmidae, this excretory system is absent or be poorly developed, but here it is developed. Mesenterial tissue is weakly developed in Phasmidae, but here it is well developed. No caudal adhesive glands is present in the Phasmidae. Okay, this is the characteristic features of the class. Now, See, now we are going to discuss about this ascaris. Ascaris. So the first thing you have to remember, as I keep on mentioning the same last two parasites, the common name. The Ascaris lubricoides is said to be a the common renown. See, Platyhelminthes, as Helminthes or the Nemathelminthes and Annelids. These three things you have to make a comparison. Okay. See, for Platyhelminthes, it is said to be a what worms. These platyhelminthes, nemathelminthes or ashelminthes, these annelids, everything is said to be as worms. Okay, they are said to be as worms. These platyhelminthes, it is also called as what? Flatworms. And this nemathelminthes is said to be as what? A roundworm. And this annelids is said to be as what? Segmented worm. And the examples for this platyhelminthes is what? Tinea solium, fasciola hepatica, like that, liver fluke and all. And this nemathelminthes, this roundworm, the best example is Ascaris. Ascaris. And for Annelida, the perfect example for Annelida is earthworm. Okay, so you have to remember which is monoecious, dioecious, and which like this. In these three phylum, which is monoecious and dioecious, and with respect to the coelom, which is true coelom, which is acoelomate, which is eucoelomate, and which is pseudocoelomate. And for based on the fertilization, which phylum is internal fertilization and which phylum is external fertilization. And when it comes to the development, which phylum is direct development and which phylum is indirect development. Anyhow, I'm going to provide you a one single page, and that will contains all the comparison with respect to all the phylums. Don't worry, but you have to focus on these three phylum particularly. This is flatworms, roundworm, and segmented worm. Okay, and this Ascaris lubricoides is the common name for this Ascaris is common roundworm. Okay, and this is the systematic position. Phylum Nematoda, Pasmidae, Ascaridae. Okay, Ascarinae, Ascaridae, Ascaris lubricoids. See, this is the one we are going to. This is the species name, Ascaris lubricoids. Okay, so you should know there are two different species of Ascariasis. And what is the species? Ascaris lubricoids and Ascaris suum. And this Ascaris lubricoids, they are going to infect the humans. Okay, and this ascaris it is going to affect mostly the pigs. See the difference between these two species. Okay, fine. See, this is the infected larva. This is the infective larval stage. Now this infective larval stages, it has been ingested with the food to the human. Now it has been reached the, anyhow they are going to reach the intestine. After reaching the intestine, it is migrate to the lungs, from where they are getting roughed up and coughed up and swallowed again. From the intestine, they are going to reach the lungs. See the final development in the small bowel. From the small intestine, lots and lots of eggs will be released into the soil through the fecal matter. For these kinds of infection, this open defecation has to be prohibited. See, right from the first class or second class, I have informed you, because of this infection, the government keep on stressing in the open defecation. So you have to aware about the open defecation. These are all the complications at the end of is as a result of this open defecation. Okay, see this by human fecal matter, the eggs will be reach the soil. Okay, and after this egg, it is going to, and initially it is going to be unfertilized the egg with a covering called capsule. Now with the help of the male or female, it is going to fertilize. It is said to be a zygote, and after the egg, it is going to hatch out into the larval stage, and this larval stage is an infective in nature. Again the cycle continues. This is how life cycle works. Now see, this is the fertilized egg passed in the feces. Okay, the eggs in the fecal matter, which has been the eggs will be hatched out into the larval stages within the egg in the soil. The egg will be the larval stages will be hatched out when they in the soil itself, and from the soil ingests the man will ingest this eggs in the food or the soil. Okay, it will reaches the mouth and it will reaches the intestine. After the intestine, it will in the venous system. Venous means veins. Okay, through the veins, it will reach the lungs, and after the lungs, it will reach the trachea, and after the trachea, it will reach the esophagus, and the esophagus, it will have the adult worms, and from the esophagus, it will reach the intestine, where the sexual maturation will be taking place, male and the female. And after the eggs and female will lay out large and lots of eggs, it will it will release the egg through the fecal matter. Again, the cycle continues. Okay, this is what happening here. Now we are going to I'm going to explain the detailed information of this. Okay, we can finish this now. Okay, first, copulation and fertilization. Definitely, it is going to take place in the small intestine of the host. That is man. Man is their host, where the adult forms live. See, so the inhabitation place, the place where the ascaris will inhabit is the small intestine of the man. The man orients its body at right angles to that of the female to copulation to be happen. See, this is the female. Consider this as the female. Consider this is the reproductive organ which is present in the female. So the male will be like this. Okay, or the male will be like this and the female will be like this. Okay, like this it will be reached. Copulation means fusion of male and female. Okay, they are getting to they are getting ready to reproduce. Sexual reproduction. Copulation means the male will reach the female and the further process will occur. Clear? They are getting ready to sexual reproduction. Okay, they are getting attached with the see to reproduce sexually. The male has to reach the female. It has to insert the sperm to the exactly. Yes. To reproduce, the male has to inject its sperm to the ovum through the vagina of the female. For that, it has to attach to, right? Okay. Yes. Okay. The male orients his body at right angles to that of female in such a way through its cloacal aperture opposite the vulva of the female. Okay. Through this vulva, the male will inject its sperm. The pineal setae, these are all the reproductive organs, okay, through which these are all the structures will help the male to reproduce with a female. Pineal setae of the male, this is one of the structure present in the male, helps to open the vulva. Vulva is the structure present in the female. This is the pineal setae is the one going to open the vulva of the female, and from the male, after it has been opened the vulva of the female, the sperms which is present inside the male, it has been transformed into the vagina. From there passes through the proximal ends of the uterus. Okay, and from once it has been entered into the female, the egg has to reach the place where the further development has to take place. And what is the safe place to hatch out those eggs means uterus. Fertilization, which is effected by the entry of sperm into the ova, takes place in the proximal ends of the uterus or the distal ends of the oviducts. After fertilization, what is fertilization? See, that scientific, what we can say, the technical definition for fertilization is the fusion of male and female gamete. Okay, that is fertilization. And as a result of fertilization, what is going to synthesize? A zygote will be produced. Okay, male and female gamete will be fused together to form a zygote. And zygote, it is an unfertilized egg, contains globules of glycogen. See, unfertilized eggs contains globules of glycogen and fat. Okay, immediately after fertilization, that is now the fusion will be occurring. Male and female gametes will be getting fused. This glycogen and globules migrate to the surface and form a fertilization membrane. Before fusion, that is before fertilization, there is a glycogen and the fat is present. But after fertilization, this glycogen and the fat, they have been converted into the fertilization membrane. Why it has to form such membrane means it has to cover, it has to protect the contents which is present inside the egg. Okay, so it is fertilization membrane which hardens into thick, clear and the chitinous shell. Okay. Now, this shell is the one going to prevent the zygote from an external danger. Next, the fat globules form a thin lipid layer below the shell. Below the shell, again the lipid layer will be formed. As the fertilized egg or the zygote moves down to the uterus, now the uterine wall secretes around it a thick, hard, yellow or brown albuminous coat. Albumin is nothing but a protein. Again, for the protection mechanism. Fertilize the egg at this stage, it looks like elliptical in nature. Okay. Now, these are all the things or an extra information. There is no need to read. For the sake of information, let me explain. Okay. Uteri of the single mature female may contains about approximately 27 million eggs with an average daily production of 2 lakhs eggs. See, these leave a mother body through the gonopore into the host intestine and finally pass through the host fecal matter. Shelled eggs are remarkably resistant and remains alive in the moist soil for several years under the adverse condition. Only because of this shelled eggs. See, these coverings are only to make the cell or the zygote to make alive for several years. Okay, only because of the cell dx these can be live for several years under suitable condition of the temperature, moisture and oxygen, and eggs undergo cleavage and develop into the infection stage. Clear? And the optimum temperature for the development is 85 degree Fahrenheit. Below 60 degree Fahrenheit, the development stops, and above 100 degrees Fahrenheit, the eggs gradually degenerate. So, what is the important point you have to remember from this slide is the temperature plays a major role, or the temperature is a deciding factor for the egg for the further process, for the further metamorphosis process. Now, how the development will be start. The cleavage here is spiral and determinate development will be here. And after the fertilization, zygote will be formed and is covered by many coverings. And after the zygote, the blastula stage will be formed after cleavage. After zygote, cleavage will be after the cleavage, it reaches the blastula stage. And what is the type of cleavage here? Spiral and determinate cleavage. Now, the eggs undergo gastrulation by invagination and finally develops into the rhabditoid stage. The most important point. You have to remember. See, what are all the points I have said above? There is no need to remember all these things, but you have to remember this stage. The question can be straight away thrown at you. What is the rhabditoid stage? Is the stage of which parasite and which phylum? Like that, they can be asked. Rhabditoid stage is the stage which has been seen in the Ascaris, okay, which comes under the phylum Helminthes or as Helminthes. Okay, it is an older name and it is a newer name. That's all the difference between this as Helminthes and Helminthes. And after 10 to 14 days, it requires to reach the second stage of larva. The hatching and further development of the larva is possible if the egg enters the host. See, the rhabditoid stage is the larval stage after the egg has been hatching out. And from the further, from the rhabditoid stage, the next stage of larva, the second stage of larva has been happened only when it reaches the host, that is human. Clear? Infection to the new host. There is no intermediate host. Is there? From the soil, human will be getting infected. Human through the fecal matter, again the egg will be laid in the soil, and from the soil, again the human will be getting affected. Clear? Is there any intermediate host from the soil to the human is seen here? There is no intermediate host from the soil to the human. From the soil to the human, from the soil to the human. Okay, there is no intermediate host, so you have to remember. So this Ascaris is monogenetic or digenetic? This Ascaris is monogenetic or digenetic? Obviously, it is monogenetic in nature. It requires only one host to complete their life cycle. Clear? In now, acquires infection. This man acquires infection directly by ingesting the Ascaris eggs containing the infective second stage rhabditoid larva with contaminated food or the water. In the small intestine, by the action of the host digestive juices, See, in the small intestine, there are many different types of alkaline juices will be secreted to digest the food. By the action of those alkaline juices, these egg shells will be dissolved. The eggs will be covered by many protective shells, and those protective shells will be destroyed or degenerated by the alkaline juices secreted by the small intestine of the human. And the juvenile will be hatched out, the second stage larva. After the shell will be lost, the juveniles will be hatched out. That is the second stage larva. And the later development and the migration. At hatching, the juvenile measures, this is the length of the juvenile, 13 mu to 15 mu in diameter. It performs active thrashing movements and bores through the epithelium of the host intestine and starts its migration in the host body. So the second stage larva is the one going to start migration inside the host body. And we have saying the second primary migration is there and the secondary migration is there. Now, the larva enters the hepatic portal circulation, which carries into the liver. As the name suggests, hepatic portal. As I mentioned before, the hepatic or hepato, everything will be deals with what? Liver. Through the hepatic portal way, it will reach the liver. From the liver, it is going to reach the heart through postcaval vein. Okay, from the intestine, it will reach the liver, and from the liver, it is going to reach the heart through the postcaval vein, and from the heart, it is going to reach the lungs via the pulmonary artery. Pulmonary artery is the one going to carry the deoxygenated blood from the lungs to the heart. Okay, by carrying those deoxygenated blood, this from the heart, it also carries the parasite from the heart to the lungs. The larva generally remains in the lungs for few days and increase in their size. Then it ruptures out the blood capillary, finally bores its way into the alveoli, where the exact exchange of gases will be happen. After about 6 days stays there, the second stage larva molds to become the third stage larva. Clear? And after the fourth day, it results in the fourth stage larva, which has by this time grown into the length of 2 to 3 millimeter. So you have to understand there are different types of larval stages seen in this type of ascaris. Okay, now the secondary migration. This is the primary migration now where they are being having in the alveoli. In the secondary migration, fourth stage larva leaves its, see what we have seen here, it reaches the alveoli and fourth stage larva will be produced by the primary migration itself. So, in the secondary migration, this fourth stage larva leaves its temporary home, that is alveoli, through the tracheae reaches the pharynx. Now, where it is going to reach? In the pharynx, from where it is coughed up and then swallowed for the second time into the intestine. Okay, from in the intestine, it molds for the fourth and the last time to become an adult. See, initially where it has been in the intestine. In the intestine, where it has been in the liver through the hepatic portal vein. From the liver, it is going to reach the heart. From the heart, it is going to reach the lungs. From the lungs, it will reach the alveoli. And from the alveoli, this is up to the primary migration. Up to this fourth stage larva will be produced, and primary migration, the end place where the primary migration will be ending is alveoli. Now the secondary migration will be starting from the alveoli. It will reach as the pharynx. When it reaches in the tracheae, pharynx, and after the pharynx, it will once again enter into the intestine. This is what the secondary migration. And when it reaches the intestine, during their second migration, they have been becomes an adult. Clear? Now, what is their end product? Adult. In the primary migration, what is the end product? Fourth larval stages. And the secondary migration, when it reaches the intestine, they'll become the adult. Okay, these are all the significance. Now, the adult, as it develops into an adult, it attains the sexual maturity within the 8 to 10 weeks. The average lifespan of Ascaris lubricans in the host is 9 to 12 months. Now, after the primary and secondary migration, the aberrant migration will be happen. Sometimes the larva of Ascaris does not follow its usual migration path but reaches the brain or the spinal cord or any such organ. Fourth larval stages means see, I have said you these young eggs will be hatched out into larval stages 1, larval stages 2, larval stages 3, larval stages 4, and later they'll become adult. And we can consider them as a miracidium, Cercaria, sporocyst, radia larva. Okay, the name of the larval stages will be different. See, we have studied before parasite. What is the name of the parasite? Cystosoma. Cystosoma, we have seen the miracidium. After the miracidium, what we have seen? Sporocyst. In that, after the sporocyst, Cercaria will be present. So this is the first larval stage. This is the second larval stage. This is the third larval stage. This is like that. After the Cercaria, in some of the parasite, radia larva will also be there, but not in the cystosoma. But generally, miracidium, sporosis, Cercaria, radia larva will be there. This is the first larval stage. This is the second, like in the same way, we are saying this is the fourth stage larva. Clear? Okay, good, good. Now, see, this is the primary migration and this is the secondary migration. But what is aberrant migration? See, as I have mentioned, from the intestine, they are going to reach the liver, after liver, heart, after heart, it's going to reach the lungs, and after the lungs, it is going to reach the alveoli. From the alveoli, again, it is going to reach the intestine. This is the usual migration path followed by this particular parasite, that is Ascaris. But in case of aberrant migration, they do not follow this usual migration, but they are unfortunately reaching the brain or the spinal cord, okay, or any other region apart from the above said migration part. That is said to be as the aberrant migration. Now, the larva is not able to survive in these organs, and the calcareous cyst will be formed around it. Okay, see, if it not follow the usual migration path like in the primary and secondary migration, it will reach the brain or the spinal cord or any other organ apart from those organs which have been mentioned in the primary and the secondary migration. If it reaches these particular organs, they can't survive. Okay, these larvae can't survive. They can't survive, and what they are going to form? Calcareous cyst. Again, during the unfavorable condition, what the microorganisms will do? They are going to cover their body parts with the extra shell, that is nothing but the cyst. Okay, when unfortunately they'll reach the intestine, again during any of the time during the favorable condition, again this calcareous cyst will reach the intestine. Once again, the cyst wall will be break down by the alkaline juices secreted by those intestine. Again the process will be continued. Okay, if it deviate from this usual path, then it is said to be as a aberrant migration. Clear? Parasitic adaptation to the ascaris. Now, how they are going to cause infection to the host? These are all be mentioned in here. What is a parasitic ascaris? Parasitic adaptation is cuticle. What is the purpose of this particular cuticle? They are going to cover the body. They are going to protect their body from an external danger. Okay, and this cuticle, how it can be prevented from the external danger means, see, in the host body or immune system or whatever it is, the stomach or the intestine, they are in the stomach. We have a proteinaceous enzyme called pepsin, trypsin, like that. These are the proteins will digest those cuticle, digest those body of the organism. So to prevent from those digestive enzymes and antitoxins, this cuticle acts as a protective shield to that particular parasite. And Ascaris also secretes some of the anti-enzymes that protect us from the host digestive enzymes. So this is the first point. To prevent from the digestive enzyme secreted by the host body, this cuticle will will be act as a protective shield for that parasite, Ascaris. Power of locomotion counteracts the peristalsis of the host intestine and compensates for the absence of adhesive suckers and the spine. See, once it has been entered into the body of the host, it's lost its tail. Okay, it's lost its locomotory organelle, and by the peristaltic movement of the stomach or the digestive tract, which is present in the human body, because of this peristaltic movement, even though the absence of suckers and spines, the ascaris can be moved from one place to another. Now, Muscular pharynx facilitates the ingestion of food by the sucking action. See, pharynx is the digestive tract organelle, and because of this structure, the parasite can be easily swallowed up once again to the intestine, and there is a continuous supply of food as a result of alimentary tract is simple without provision of storage. It is easy for ascaris to survive in those environment. Ingested food is pre-digested so that there is no elaborate digestive glands. Pseudocoelomic fluid serves as an absorption, transport, distribution of food, oxygen, waste. So there is no need of circulatory system. Even though there is no need of circulatory system, the functions performing by this circulatory system has been done by some of the structures which is present in the ascaris. So there is no need of circulatory system. Extremely low metabolic rate. Okay, see, low metabolic rate because there is an anaerobic respiration. See, in case of oxygen is present, we can said as the aerobic respiration. If oxygen is present, the metabolism rate of metabolism will be higher. But here, low metabolic rate because of the absence of oxygen. So there is no need of respiratory organs too. This enables the worms to live inside the host intestine where the free oxygen will be negligible. As this ascaris will prefer to live in the anaerobic respiration, they are going to prefer to live in the intestine. Why the every organisms prefer to live in the gut region of the host means because there is no need of oxygen there. Okay, even though the metabolism rate is low, they can manage with those particular environments. So they prefer to live in the gut or the intestine of the host. Parasite remains well protected inside the host intestine from predators so that there is no need of complicated sense organs. Okay, there is no complicated sense organs will be necessary. Instead, the sensory organs will be very primitive. That is sufficient for the parasite to survive. The resistant covering or the shell provides safety to the zygotes. Again, this is going to prevent in the adverse condition or the unfavorable environmental factors. Now, minute size, resistant nature of ducts afford far and the wide dispersal of the parasite. As it has a resistance with it, as it has a protective covering, as it has size is very much lower, it can be easily dispersed through a larger surface area. Even though the longer distance, it can easily dispersed because of the size is very smaller. Infection to the new human is direct. There is no intermediate host. So it is very favorable for those particular parasites where the no, it doesn't want to wait for the intermediate host to carry those eggs. It doesn't wait for the primary host from the intermediate host to the primary host. So it is very direct. So it is very fast for those parasites to enter into the body of the host. Now, what is the name of the diseases caused by this Ascaris lumbricoides? Ascariasis. They are going to cause the infection. Wait, this is the name of the disease caused by the Ascariasis is Ascariasis. How they can be transformed? By consuming the food and water contaminated with infective eggs. So both food and water will act as the mode of transmission, and it is this the incidence of infection will be greater in humans than in the adults because they play in the grounds, they play in the infected water and infected soils, and also they are become more vulnerable than the adults. What are all the pathogenesis? What are all the diseases they are? What are the symptoms going to cause in this particular disease of caries? Is causing hemorrhages, blood related diseases, and they sometimes go through the intestinal epithelium, they enter into the circulation, and they reach some of the important organs like kidneys, spinal cord, brains, or muscles. They cause serious injuries. Okay, if they reach the brain, it can even cause the paralytic conditions also. Okay, now severe hemorrhagic conditions develops in the lungs, which causes the petechial hemorrhages and inflammation of alveolar tissue followed by the edema. These are all the hemorrhagic conditions. In severe infection, they may cause acute pneumonia. Okay, acute pneumonia with fatal consequences. And frequently, this infection is immediately followed by temperature, anemia, and leukocytosis eosinophilia. These are all the conditions caused by the ascaris. Now, also they can migrate into the enterites. Enterites means related to the intestine, small intestine and large intestine. Migrate into the vermiform appendix. In intestine, we have a small elongated structure which is called as the appendix. It looks like a worm-like structure. Vermiform is a worm, and so it is called as a vermiform appendix. So these parasite will reach the vermiform appendix, gallbladder, bile duct, so they can cause the inflammation. This adult parasite will draw their nourishment from the contents of the intestine. We know that. Now, they can cause symptoms, some of the convolutions in the eyes, delirium, and nervousness. Everything will be happening when we are getting infected with this ascaris. And these are all the things are not that much necessary. Okay, so don't need to no need to reach that. And what are all the drugs going to treat this particular ascaris is anti-helminthe drugs. And what is the anti-helminthe drugs? Chenopodium. Oil of chenopodium is very effective. And most modern successful anti-helminthe drugs is hetrazine and piperazine hydrate. Okay, if you have any doubts, you can ask me now. If you have any doubts, just say me yes or no. In tomorrow's class, we are going to deal with this Ancylostoma trichinella, okay, and LOA. These three things we are going to discuss by tomorrow. Okay, I think this will take further exactly 2 class. We need 2 class to finish, 2 or 2.5 class to finish this Helminthes, and after that we'll move on to further phylums. Okay, any doubts? If you have no doubts, you can leave the class now. Please be regular to the class, please be on time. Okay, fine. Thank you. Thank you.